Okay, maybe we can get started. So hello, everyone. My name is Andrej Kosmrlj, and I'm assistant professor in the Department of Mechanical and Aerospace Engineering at Princeton. Today, I'll give you a lecture on multicellular structure formation in 3D. And if you're wondering what are these two images, so on the left, you're seeing the image of the wrinkled bacterial biofilm. And on the right, you're seeing like a section of the mouse lung. And on the right, you're seeing like smooth muscles, which are wrapping these lungs. And we're gonna talk a lot more about these two projects in the second part of the lecture. But in the first part of the lecture, I just wanna give you like some general ideas of how we can form multicellular structures, right? And so if you look around in nature, we're seeing like lots of different types of structures, right? So we're seeing like helices, spirals, like we're seeing that like lots of animals have like stripes or spots, like we're seeing like wrinkles in brains, or like, you know, like we're seeing like these like little pillars in guts, or like we're seeing like branched networks in lungs and kidneys, and so on. So there are like lots of different, and you know, we're seeing more and more spirals in plants. And I'm also going to mention a little bit about structural colors and about the regenerative properties of the hydra. And so, you know, like this diversity, biodiversity has really inspired like a lot of scientists throughout the history. And like about like more than 100 years ago, there was like a very inspiring book that was written by Darcy Thompson with the title On Growth and Form. And in this book, Darcy Thompson really like tried to provide some mathematical formalism of how we can describe different types of shapes and forms, and also like how different types of growth profiles can give rise to different types of form. So in the next few slides, I just want to give you like a brief overview of how different types of mechanisms can give rise to these structures. And by no means this will be a complete list, right? So I will just want to give you like some ideas, and there's like obviously a lot more mechanisms that are out there. Okay, so we're gonna start first with like helices and spirals, right? So I already mentioned that, like, you know, like in plants, we often see helices. So here you're seeing like a helices in the vine tendril. Last week, Professor Shevitz gave in his lecture mentions bacterium, right? So these bacteria have flagella, which act like propellers. And even these flagella, they look like a helix. And, you know, but in nature, we also know like there are like, we have like lots of examples where we're seeing spirals, such as shells of snails, beaks, claws, horns, teeth, tusks, and so on. And like, and these, all sorts of these different structures can be obtained via very simple growth rules, right? So imagine that we have like if we imagine that we have like a 2-layer rod, right? And if one rod grows faster and one side of the rod grows faster than the other, the result, because this blue layer will get longer than the red one, this beam will curve, right? And if the beam becomes very long, then this is going to curve onto itself, and then it's going to form like this helix, right? And here I'm talking about the faster growth of one layer. Sometimes it also happens that in response to some external cues, you know, the red layer may shrink relative to the blue, right? So whether one layer expands or the other one shrinks will be equivalent in this scenario, right? And if you have like just a uniform growth of one layer with respect to the others, then typically you're going to get like a constant curvature. And if the beam is very, very long, then it's going to form like a helix, right? And if you want to get like a spiral, then it's not enough just to have like a differential growth between the two sides, but also the width has to grow, right? And if you grow these the two sides and the width with the constant velocity, so like, you know, like, so here, like in time delta t, you're adding like a material v out times delta t, here v in times delta t, and here vw times delta t. So if these growth velocities are constant in time, then what you're going to end up is like, you're going to end up like with these spirals. And indeed, like quite often in all of these examples, it's only the outer, like the outer part of the spiral that's growing, or in these peaks. And sometimes, so, you know, most of the part, you're going to add up new material at the ends. And sometimes you can also add new material all the way on the surface, right? But the idea is still the same, that like if you're adding more and more material on the top and the bottom, and then also through the thickness, then you're going to start creating spirals. In nature, we also often see wrinkles. For example, you know that if you're taking a bath, right, then your fingers will get wrinkles. You know, our fruits, if they become, if we don't eat them for a long time, they dry out and become wrinkled. But like all of these wrinkles, they also show up uh, in development biology, right? So, for example, here we're looking at the wrinkling of the brains, and here we're looking, here we're looking at the villi inside the gut. And I'm going to explain you that during this, that the formation of villi is actually preceded by these wrinkles, right? So, why do these wrinkles actually happen, right? So maybe it's the easiest to think about what's happening in apple. So, apple has the internal fruit, and then it has like a skin on the outside. But as it dries out, but as it becomes older and older, like it's basically losing water. So the interior shrinks, and now the surface area of the skin is too large for a given volume. So in order to accommodate this excess area, it's going to create these wrinkles, right? And something similar is happening when we take a bath. So basically, what's happening, there's like a physiological response that our veins inside will constrict. So again, just like in apple, the interior will shrink, and now the surface area of the skin is too large for a given volume. So again, it's going to accommodate these wrinkles, right? And and I'm going to talk like later in the second part of the lecture what actually sets the wavelength of these wrinkles, because you see that like the wavelength is quite uniform, right? On the other, so you know, here in both of these cases, it was the interior that was shrinking, but it's actually something different that's happening in the brain, right? So again, all the wrinkles that we get, basically most of the wrinkles that we have, we get them before we are born. So these are the MRI images of the brains of the human fetus. That was like 20 weeks old, that was 20 weeks of the pregnancy all the way up to 36, 37, just before the fetus was born. And what you're seeing that initially the surface is smooth, but then it started to get more and more wrinkled. And what's happening that during this stage, after like halfway through the pregnancy, the neurons start migrating from the interior towards the cortex. And as a result, the surface effectively swells. And the net effect is the same as here. The surface area becomes too large for a given volume. And in order to accommodate excess area, the surface will wrinkle, right? So, you know, like here it was the interior that was shrinking, but here it's the surface area that's expanding. And inside the gut, and then the reason why we want to have like this wrinkles so that we can pack more and more neurons, because we use them for thinking. Inside the gut, we have like these little pillars, which are called villi. And we have like these pillars so that we effectively, so inside the gut lumen, so we effectively want to increase the surface area of the interior gut tube so that, you know, like the processed food can get absorbed by these villi, and this will then be sent to these nutrients will then be sent to the bloodstream, right? And what you're looking at here is these, so these are the cross sections of the guts in chicken embryos that was 6 days old, 12 days old, 15 and 17. And this is the cross section like this. And this is showing what's happening on the interior surface of the lumen. So again, you see that initially it's smooth, just like it was in brain, but eventually you start seeing these stripe forming. And if you look at the cross section, you see that these are undulations that are going up and down, right? And these are indeed just wrinkles, just like here. And the reason why these wrinkles formed is like, if you look at these cross sections, so here the blue color just marks all the cells, but the green color marks the muscles, right? And outside these gut tube, there are like two types of muscles that form. There are longitudinal muscles, which are these stripes, and circular muscles, which go around like that. And what's happening in the first part at day 12, the system is starting to form circular muscles. And these muscles are actually stiffer than the other tissue. And muscles, they also grow slower than the other tissue. So these muscles are effectively compressing the tissue like that, in that direction. So they're effectively compressing in the circle direction, which is going to create these stripes. Like a few days later, what's happening that at day 15, the chick embryo also starts forming longitudinal muscles, which will again grow slower than the rest of the tissue. So the system will now not be compressed just in the circle direction, but also in longitudinal direction. And now, instead of getting straight stripes, we're going to get these zigzags. But these are both wrinkles, just like here, right? And then what's happening, and then we're going to talk more about these straight versus zigzag wrinkles. And then what's happening that Then these zigzags then serves as a template on which these villi, these bumps, will then grow, right? So again, like these simple wrinkling instabilities can be used to serve to form these various kinds of patterns. Okay, but here, like in both cases, on the previous slide and on this slide, the growth patterns were very simple. Like there was like one layer that was growing faster than the other, and in one case, this gave rise to helices and spirals, and if the substrate was thick, then this gave rise to wrinkles, right? But sometimes we also want to have like a more complicated growth patterns, right? So now we need to figure out, so now the system has to figure out how to coordinate these different growth patterns, right? And this is often done through some chemical signals, which was not, and this is usually dubbed Turing patterns, right? And this is named after Alan Turing, who wrote like this very influential paper in the 1950s, right? So as I mentioned, like often in nature, like, you know, especially in animals, we're seeing like animals with like stripes and spots. Uh, but you can also find them like on a larger scale. For example, like these are so-called tiger bushes, which you can find when, again, you see that you kind of have like some patterns between regions where you have a bush in green and regions where you don't have a bush. And then also you can find them in forests. So if you go very high to the mountains, you can, so this is the forest made of fir wood, and you can see that like the density oscillates. So again, and you can even find them on beaches. So these are like patterns of mussel shells, mussels, and then like even like hallucination patterns can be described with the same formalism. And Alan Turing figured out that like all of that, like many of these patterns can be described with reaction diffusion-like equations, where Ci are the so Ci would be concentration field of molecule of type I at position R in space and time t. And then this concentration, and then, you know, like if you have like two components, I will be 1 or 2. If you have more than two components, then I will go from 1 to n, where n is the number of components. And then each concentration field will evolve in time like this, where you have like two terms. The first are some functions that depend on the local concentrations of all the molecules. And then these functions were basically telling like how these different molecules, how they interact with each other in order to produce or reduce the number of different types of molecules. And then the second part is telling you something about the diffusion of these molecules, where the I are the diffusion coefficients. And what Alan Turing figured out, that these simple sets of reaction diffusion equations can give rise to patterns, right? And here I'm going to show like a simulation on top for a two-component system that's going to give rise to stripes, right? So where the color is just indicating the concentration of one of the molecules. And if I change these functions, you know, like I could get spots, but I can also get stripes, right? And what you notice here that like over time, this pattern, you know, just became static. And this is typically what happens when you have two components. And even this is also what happens often when you have more components. But if you have three components, then besides having static patterns, then you can also get these dynamic patterns, which you're seeing here. And these are like, and the prototypical example is the so-called busy reaction, which is well known in chemistry. And while the reaction diffusion equations of these Turing type can indeed give rise to these patterns, this doesn't mean that actually all of these patterns indeed come from the reaction diffusion like equations of molecules. There are definitely also instances where patterns can arise from other mechanisms, but indeed this was also like a turning point. This paper by Alan Turing was a turning point which gave biologists to start thinking about these patterns in a completely different way. Okay, so the reason why I mentioned these chemical patterns is because quite often chemical patterns then can guide the growth of cells, right? So like in our body, like we have like a lots of networks and branch structures. For example, like the nervous system is like, you know, like a very huge network, the vascular network. And then like we also have like lots of branch system. So here's like an example of, in this blue, you see a respiratory system of Drosophie, which is like a fruit fly. Here you're seeing like branched structure of the mammary gland, branches of kidney, and also branches of lungs, right? And quite often what is happening is that, again, like through these chemicals, which are highlighted here in red, and here you have like an actual name of which sort of like molecules are diffusing around. They sort of like through this reaction diffusion type process, they form patterns, and then cells can sense these molecules and they respond to these molecules, right? And, you know, like depending on the types of the structure, you know, like sometimes these signals, they will cause the cells to migrate toward the signal, such as is the case in nervous system and vascular networks. Sometimes they can cause the cells to start dividing more, which is the case in the mammary gland. And sometimes what's going to happen that these signals are going to cause constriction of some cells. For example, like here, what we'll see later. Also like in mammary glands, you have migration. In kidneys, you have proliferation. And in lungs, what will happen later is you form these smooth muscles, which provide constriction. And this is what happens in mammalian lungs. Whereas in like bird lungs, what happens is that actually these cells constrict. But the main idea is that there's some interaction between like a chemical signals and the growth and the proper behavior of cells. And as Matt mentioned this morning, there's like also like a feedback because the state of the cell, if the cell is under mechanical stress, it can also affect the properties of the signal. So there's always like a feedback loop between the chemical signals and the properties of the cells. Okay, now I'm going to show like another example, which is very fascinating, of how this interaction between chemical signals and formation of new cells gives rise to very interesting stripes, right? So like in a lot of plants, if you look at the leaf patterns, right, you're going to see some stripes like this, right? So this is like, so these are like spirals of the aloe plant. And like what spirals we're talking about is like, for example, if you're, and then also like here, you're very clearly seeing these right-handed spirals, but in fact, there are also like a left-handed spirals, which maybe are a bit harder to see, right? But here I marked them on the case of the sunflower, right? So here you see these, so each of these white line is like a different spiral, and these are left-handed spirals, and then these are the right-handed spirals for the same image. And what is very fascinating, if you count how many spirals there are, you're going to see that there are 21 in here, and then 34 in here. And if you count all of them also for these other images here, you'll see that you have like here 2 and 3 spirals. So one number tells you the number of left-handed, and one number the numbers of right-handed spirals. And which one is left, which one is right can be different. Right? But what is interesting is that these numbers 2, 3, 5, 8, 13, 21, 34, and 55, they form a Fibonacci sequence, right? Which is very interesting, right? So why on earth would like these plants create like these spirals, and the number of spirals like, you know, forms the Fibonacci sequence? So this was like a very interesting puzzle. And like eventually it was figured out that what's happening is that like how all of these leaves are made, they all grow from the shoot apical meristem, which you're seeing here. And if you zoom in, what you're seeing is that like you're seeing these little buds. So these are the primordia, so-called primordia, from which you're going to eventually create like all of these leaves and petals and so on. Right, and then like these numbers here tell you like at what stage this primordium was generated. So 1 is the most recent one, 2 is the one that's generated just before, 3 just before, and so on. And what's happening is that like once each of these primordium is generated, it releases some hormones called auxin, and these hormones are now diffusing around. They eventually decay, but what happens is that the new primordium wants to will appear at the site where it's depleted of these hormones, right? So so like very close to this number 2, there'll be like a lot of auxins, right? So 1 would not like to form near 2. And also near 3, there will be a lot of auxins. So 1 would not like to form near 3. And then over time, this, like amount of hormone will degrade, right? So like maybe 10, there was initially a lot of hormones here, but over time these degrades, right? So this will get depleted. So eventually, like one would like to form as far away as possible from the new ones, 2 and 3, and then eventually, like if you then measure these angles, very fascinatingly, like eventually, like they will form like these angles will form so-called golden angles, right? And if you continue this process, what you eventually find, like that was done in some research literature, that like eventually, like in this system, if you have like like these, if you think of this as disks, they're repelling each other, you're gonna start getting these Fibonacci spirals. And these spirals are happening in the center. I mean, these Fibonacci numbers are happening in the center with spirals, but also if you count the number of petals, you will see that they also, the number of petals is also equal to one of these Fibonacci numbers, which is very interesting. Okay, another process that's helping to produce these patterns is so-called phase separation. You probably know that if you pour oil in water, oil will not mix with water, so it will stay separated as an oil. And then eventually, because of the buoyancy, because the oil is lighter than water, eventually it's gonna all come on top, right? And the reason why this happens is because oil molecules like to interact with each other more than with the water, right? So the energy can be minimized if the water molecules are close to each other, but they're not close to the water molecules. And something very similar is also happening in biology. And this was actually like, and for example, here what you want to see is an example of so-called intracellular condensates, right? So inside the cell, we have like a lot of these different organelles. And for a long time, biologists thought that they're really separated by each other via membrane. But in the last decade, it was recognized there are also organelles which don't have membranes, and they form via phase separation, just like between oil and water. And here I'm going to show you like a movie where different types of molecule are labeled with green and red. And these are the condensates that are happening inside the nucleus of the frog egg, and they form, and these blobs, they're called nucleoli. And if you look at this movie, this looks just like fluid phases that are coalescing with each other. This looks just like oil droplets. They're merging and coming together, right? And since the original discovery around 10 years ago, now biologists have recognized that these fluid-like condensates appear in many places in biological systems, and they actually are playing a very important role. And another interesting place where the phase separation shows up is in the context of structural colors, right? So here you see a bird called indigo bunting, but this bird, the color of this blue color is not coming from the pigment, but is rather coming from a structure. In fact, for biology, it's very hard to create like a blue color. So the blue color, if you see blue color, it's often coming from the structural color. And the structural color here means that like if the sunlight, so like I said, the bird is not really blue, but like it appears blue to us. And the reason is the following: the sunlight, which is white, contains all of these different colors. And when it falls on these feathers, most of the colors will pass through, but the blue light will get reflected because of the constructive interference. And it's this reflected color that we see, right? And this light is this constructive interference happens on this keratin air matrix. And it was shown in this paper around 10 years ago that this keratin and air, that this structure forms via phase separation, just like we saw in oil and water and in these intracellular condensates. So it's very interesting that like this phase separation can appear in many different contexts. But the main reason is just like the standard thermodynamics. And in my group, we're also doing some research on the phase separation. And if you're interested, you're welcome of how we can, like achieve like all of these different types of morphologies in phase separation. Then I would direct you to read these two papers. And I will not talk more about these because in 2 weeks, you're going to have a lecture by Professor Ned Wingren, who's going to talk a lot more about the phase separation. So this phase separation also appears in other contexts. So here you have like an example of a cell sorting in organism called Hydra. And then this cell sorting looks, and the reason why these cells sort is just like in a phase separation, like there's like a differential adhesion. So the purple and green cells, they have like different amount of stickiness to the cells of the same type and cells of the different types. And as a result, they phase separate. And this Hydra organism is fascinating because it can self-regenerate. For example, if you cut the Hydra organism in half, so that here you have like a head and here you have like a tail, like a few days later, like this top part will grow a tail, whereas the bottom part will grow a head. So now I have like two organisms, right? And like what was fascinating is that like they went in this paper, they went a step further. They took the organism, like a few of them, and they, or maybe just one of them, I don't know. And then they dissociated the cells. So you now have like an aggregate of individual cells, right? Which is shown here. Over time, like these cells will sort each other so that like, the red cells here or the purple here will be on the inside and the green cells will be on the outside. And again, the mechanism is just like a phase separation that I showed you. And if you wait like a few days later, now you start seeing that the head is popping up here, another head's popping up here. So like, like you started with like a collection of cells that have sorted each other out, and now they have developed into complete organism, right? And it's thought that this cell sorting mechanism is also playing a role during the developmental stages of the embryos. Okay, so the last slide that I have before like I take a little break for questions and answers is also like I'm going to talk a process of solidification, and even a process of solidification can give rise to interesting patterns. For example, like on the left, you're seeing like a snowflakes that were imaged by Wilson Bentley like more than 100 years ago, and you know that these snowflakes, they form like these hexagonal crystals, and why they form hexagonal crystals just because of the nature of the water molecule bonds. In biology, quite often, like in natural systems, like we want to avoid solidification because it gives rise to certain diseases. Of course, like we want certain types of solid structures that give rise to cell support and cytoskeleton, but other than that, they often are associated with bad things, right? So on the top part, I'm showing the sickle cell disease. And this is coming from the following. So these red blood cells, they're carrying like a lot of hemoglobin proteins, which can bind oxygen so that they can carry around oxygen. But if these cells are in low oxygen environment, these hemoglobin proteins, they can start solidifying and they form these like long strands. And once they make these long strands, then these cells become very stiff, and they form these crescent shape. So that's why these are called sickle cells. And once they're so stiff, they cannot squeeze anymore through these like narrow capillaries, which is like a problem, right? And another example where these like solidified aggregates are causing problem is in neurodegenerative diseases, for example, in the Alzheimer's disease, right? So this is, so in the Alzheimer's disease, what's happening is that like you're getting like these plaques, like solidified plaques of amyloid fibrils. And these solidified plaques, they are toxic for the neurons. So as a result, the neurons will start dying, and you'll have like fewer neurons. And there's like another part of aggregates that can form. So inside the neurons, like you have like these long cables, which are called microtubules. And inside the microtubules, you also have so-called tau proteins, which provide like stability to these microtubules. And these microtubules, at least in the context of neurons, they participate during the communication between neurons, right? Because the neuron has to send a signal along these microtubules to another neuron. But if the tau proteins, if they form tangles with each other, they will not bind anymore to these microtubules, which will lead to disintegration of microtubules, which will also will disrupt communication between different neurons. Right? So both of these plaques effectively lead to a loss of neurons. So that's why if you look at the brain of these patients with the Alzheimer's disease, you will see that because of the loss of neurons, their brains are much, much smaller compared to the normal brain. Okay, so maybe let's take a break here and I can look at some questions. Okay, so I look at the top question is there correlation between complexity of brain regions versus its level of wrinkling? Okay, so that's a very good question. Let's go to the brain. Right, so the answer is no, right? It seems that these wrinkles appear quite uniformly. Like what you may notice is that in some regions, you're starting to, like there are some regions that where you're first getting folds, and these are so-called primary folds. And these folds are quite similar between all humans, and they're dictated by the shape of the brain hemispheres, right? And then you're starting to get in like smaller and smaller folds, and the actual patterns of these small folds is not really related to any of our cognitive ability, right? Because they really are just a consequence of the fact that we have that the surface area has increased. And over time, these, like I said, most of these folds we're getting before we are born. And over time, we are not really getting, generating more folds, but maybe we are just, like as we're young, we're still adding more neurons. So the whole brains are still growing. But once we, once we become teenagers or adult, or once we reach 20s, then actually we are not forming new neurons and we're just rewiring them so that so we form new memories by rewiring neurons, but we're not really generating new neurons. Okay, and then okay, is there maybe so let's go to next question. So are there Fibonacci patterns also governed by the reaction diffusion equation? So I think I commented on that like there's like these auxin hormones, right? They're diffusing around. And yes, you could say that there's some reaction because the cells are sending the auxin, so they're also responding. So in that sense, they're also reacting to it. Okay, are there always more right-handed spiral, or is this just the case for these pictures? The question is, no, in general, there can be also more left-handed spirals than the right. Like it really depends on the plant, and even like the same plant, the handedness can be opposite. For example, if you take two sunflowers, one of them may have more left-handed spirals, one of them may have more right-handed spirals. I don't think there's anything that tells you which type there should be more, unless there's some other mechanism that breaks the symmetry, but typically there are none. What is interesting, like I said, is that these two numbers, they form two consecutive numbers of the Fibonacci numbers. Okay, maybe let's do one more question and then we can move on. The self-sorting of hydra look a lot like an effect you might see from surfactants of block copolymers. Is the underlying mechanism similar? And the answer is yes. Let's just go back to the cell sorting, right? So like I said, the cell sorting is coming from these interfacial energies, right? So it's interaction between like how strongly these molecules or cells in this case, they interact with each other, or in the block copolymers, how strongly these chains of different blocks, how they interact with each other. And it's this competition between the interaction energy between different types of molecule and the mixing entropy, right, which gives rise to this phase separation, right? And in these cases where the separation happens, the entropy is not so important. So it's really mostly in the interaction energy between here, like purple and purple, green and green, and green and purple, that dictates how they're going to sort each other out. And that's true in like a passive phase separating system, in self-sorting systems, and also in block copolymers. Okay, so now let's move on so that we have some more time for other parts. So now, first, I'm going to briefly comment about the nature of the wrinkling instability on the origin and what sets the wavelength. Then I'm going to comment on the wrinkling of the bacterial biofilm, which was done in collaboration with Bonnie Basler, Howard Stone, and Ned Dingreen here at Princeton. And then I'm also going to comment about the branching of the developing lungs, which was done together with Celeste Nelson, also here at Princeton, right? As we already said, the wrinkling typically happens when you have like a thin skin on a soft substrate. And it's importantly that this happens when the skin is growing faster than the substrate. So this factor GF and GS will tell you by what fraction did the length increase after growth for the film, and GS, what fraction did the length increase by the substrate. And if we force this skin and substrate to be bonded together, then if the mismatch of growth is smaller than some critical value, then the film will stay flat. But if the mismatch in growth is larger than this critical threshold, then it's going to form wrinkles. And interestingly, if the film continues to grow relative to the substrate, then the wavelength will stay constant, but the amplitude will increase. So this plot here is showing the amplitude. And while the wavelength is set by the stiffness of the film and the substrate to the 1/3 power and to the thickness of the film, right? And this was already known like over half a century ago, and you can find this in the book by Allen, right? And like I said, the amplitude continues to grow like in a square root fashion, right? So now I'm gonna go outside presentation and I'm gonna show you like a video. So here I have like an example of such system, right? What we're seeing here is like we have like a thin skin, which is like gray, which is stiffer than the soft substrate. And here, like I'm not gonna grow the film, but I'm gonna compress the film by bending it, right? So if I bend it upwards, then the film gets compressed. And if I bend it downwards, then the film gets stretched. So notice that here, what I was asking is that the film goes faster than the substrate because the wrinkle happens. If the substrate would go faster than the film, then the film would have to stretch like here and nothing happens, right? But if you observe what happens is I'm bending it, so now the film is getting compressed. If I bend it gently, nothing, then initially nothing happens. But if I bend it enough, then I'm getting these stripes, which are these wrinkles that I'm showing here. And if you look from the side view, you see that like once the wrinkles form, you see that the film is going up and down, right? So you see, let's see if I can do this. So you're seeing that the film is going up and down, but also the pink substrate is also getting deformed. It's also getting stretched up and then compressed down. So basically what sets this wavelength is sort of like a competition between the bending of the film and the stretching of the substrate. And the reason why it wrinkles is because the compression of the film is very hard. So the film can actually release this compression by bending, which is energetically less costly. And while you don't have this sample, you can just try to play with your skin, right? So if you compress your skin gently, nothing happens. But if you compress it enough, then you're gonna start seeing wrinkles. And again, you see that wrinkles are orthogonal to the direction of stretch, of compression, right? Because they're releasing the compression in that direction. Okay, so now let's go back to the presentation. So if you're compressing in one direction, then you're gonna get stripes. But if you compress in two directions, then what's happening is that, again, the critical mismatch of growth is very similar. So here, actually, these pictures are not from growth, but these are done from the swelling experiments of hydrogel, where the red skin is swelling isotropically in all directions. So as a result, the compression is not just in one direction, but it's actually in two directions. So it's biaxial. And what you see that initially the surface is smooth when the swelling is low, just like I said on the previous slide. But once the swelling of the red layer reaches the critical threshold, which is the same as before, then first it formed like these little bumps, but eventually these bumps merge into these hexagonal stripes, and these hexagonal stripes, and these zigzag stripes, and these zigzag stripes, these are called herringbone patterns. And interestingly, if you measure the characteristic spacing between these herringbones, they scale in the same way as the stripes that are compressed in one direction, right? And I'm going to try to argue that something very similar is happening in the wrinkling of bacterial biofilm. So here we are studying the biofilm that's made of bacteria called Vibrio cholerae, and that's the same bacteria that gives like a cholera disease, right? So we grow these on like some soft agar, which produce nutrients. So we just make like a small drop containing bacteria. Each bacteria is like roughly 1 micron, like of the order like 1 micron in size. And then what is going to happen is they're going to start consuming nutrients from the agar, and they're going to start growing and they're dividing. So they start expanding, and they form like a thin pancake. So they start expanding and dividing, and after a while, you start seeing these stripes, right? And interestingly, you see like stripes, they're like pointed in the radial direction here, and while they form like these zigzag stripes in the center. And these indeed, like if you look at the 3D profile, there indeed are undulations, and we claim that these are indeed wrinkles, right? And so this biofilm forms like a thin pancake, right, which is like roughly 100 microns thick, and it's like like a few, and it can be like a centimeters in size, all right? And the reason why, why we start getting, why they start getting wrinkles is because as the cells are expanding, they also like stick to the agar substrate, so they're sticking. So when they try to expand, the agar is pushing back. So this effectively provides compression. And in the center, like, like cells are constrained everywhere. So they are feeling, so when they try to grow and expand, they're getting pushed from everywhere. So we start getting zigzags. Whereas at the leading edge, like I saw the movie, they're expanding, right? So they can release, so if they want to grow, they can release the compression in that direction by expanding, but they're still compressed in that direction because of their neighbors. So as a result, we're getting like stripes. They're like just so straight stripes here that are compressed in one direction, whereas we get zigzags here. Interesting, you see, if you look at the characteristic spacing, it looks very uniform, looks the same in here and in here. And this is because the wavelength of wrinkles only depends on the thickness of the film, which is roughly constant. And I forgot to mention, this thickness is set by the oxygen penetration depth. So these bacteria, they need nutrients from the agar, but they also need oxygen from the top. And this is what sets the thickness, right? And because the wavelength is constant, as the biofilm grows larger and larger, the perimeter increases. So you have to feed in more and more wrinkles. And you also see that you're starting to see these bifurcations where you start getting additional wrinkles, right? So this is what's happening in the early stages. So over like the first 2 days, if these biofilms continue to grow, then what happens is what you will see that some of these stripes, they become much darker than the other ones. And what happens that actually like some of these biofilm actually has delaminated from the substrate. It's not attached anymore to the substrate, but it can, because it only sticks with a finite amount of energy, it can actually further reduce the energy by detaching from the substrate, right? So definitely the morphology of these biofilms can be very interesting. And in our study, we mostly focused on the first part. And interestingly, what we notice is that if we change the geometry of the biofilm by making these thin strips, for example, by dipping a razor blade in a solution of bacteria and then just stamping these razor blade on the agar substrate, if we now grow these biofilms, what you will see that again, they start forming wrinkles. Again, the wrinkles are oriented or thought in the direction of the growth because they release the stress compression here and they're compressed in that direction. And if you measure the wavelength here and wavelength here, they're the same. And this is because in these wrinkling problems, the wavelength is not set by the geometry, but by the material properties and the thickness of the film. And we went actually further. So we actually were able to manipulate the wavelength of the biofilm by changing the stiffness of the substrate, right? So here you're seeing biofilms that are grown on different agars with different amount of agar concentration, which changes the stiffness from a fraction of kilopascals to a few kilopascals. So so these are still quite soft compared to most other materials, right? And now what we did, we measured the wavelength from these images. And then we also measured the stiffness of the film and stiffness of the substrate. And we also measured the thickness of the film. And here we plot the measured wavelength, and here we plot the predicted wavelength from the mechanics. And we indeed see that we have a very good agreement between the two, which suggests that all of these patterns that you're seeing here, they're indeed coming from the mechanical instability, but not from some other Turing-like mechanism, which would happen from the chemical reaction and diffusion. Like in the follow-up study, we also went a bit further to try to explain where we are seeing the regions of the radial stripes versus these zigzag herringbones. And in the core set, inside, you also see some a bit distinct pattern, which is basically coming from the initial drop size. and it's not really related to the mechanical instability. And we can track where these regions over time. For example, like here, this is so-called kymograph, where at different time, we plot where these regions with zigzags or stripes as a function of distance from the center, and the gray is telling you where initially when it was like smooth. Like I said, initially the bacteria, so this is not a movie, which is showing, so basically we track this movie and we track where are the stripes, where there are no patterns, and also then where the zigzag show up. So we see that the zigzag region expands over time, which is what you're seeing here, and also the stripe region expands over time, right? Interestingly, like if you grow this on a stiff substrate, the patterns first emerge in the center and then propagate outwards. But if you grow them on a soft substrates, what happens is the wrinkles first show up at the outer edge and then they propagate inwards, which is what you can see here. So here the blue region starts in the center and they propagate outwards. And here it starts at the outer edge and it propagates inwards. And in this study, we actually developed like a model where we take into account both the diffusion of nutrients from the substrate, the uptake of nutrients, by bacteria, the growth, and the mechanics of the formation to try to explain this behavior. And what we saw that the difference between these two scenarios is that here, the maximum stress, compressive stress that the bottom experiences is in the center, whereas here the maximum compressive stress is the outer edge, because bacteria basically like they eat up the nutrients in the center, so they start starving and so on, which is also the case in the upper case, but because of the higher stiffness of the substrate, they cannot really expand that much. So maybe let's take another break to answer a few more questions. Okay, so first we get a question, so why do wrinkles form on people as they get older, right? So this is again a very good question, right? So like I said, typically wrinkles happen when the skin surface area is too large for the substrate, right? And what's happening as we get older, like our skin is actually getting, so basically, like maybe you've noticed in your, like older relatives or older people that like basically like over time, their face shrinks a little bit. So they lose some of the muscles and some of, so basically the interior skin and the interior substrate shrinks, whereas the skin area remains the same. So it's the same idea as I was showing you with an apple. So the interior shrinks and then the skin area is too large, so it's gonna adopt these wrinkles. This looks like an iris. How is the pattern iris is formed? So I'm not sure what exactly this is referring to. So I'm gonna try to maybe answer these like offline afterwards. In the blue color is caused by constructive interference. Shouldn't we see the color of the bird differently depending on the angle of reflection? Okay, that's a very good question. Let's see where we are. Okay. Okay. So indeed, what happens if this matrix would be like a perfect crystal, then depending on the angle, you would see like a different color. That's correct. Sometimes what's happening, I don't really have like an image here, but sometimes these bubbles, they're not perfect crystals, but they're more like a disordered bubble. Like they have disordered positions. But if they're disordered, but they have like some characteristic spacing, then they will form the same color, reflected color, regardless of the angle, right? So that can happen. And often during the phase separation, you're going to get like a bit more disordered structures rather than the perfect crystals, which you see in some photonics. Okay. Okay, we answered that. Are there even cases in which the parasitic numbers are equal? And if so, what effects are observed when compared to biological relatives? Okay, so parasitic numbers are usually not equal. Like you always have, like they always correspond to two consecutive numbers. Two consecutive numbers of the Fibonacci sequence, right? So they're not equal, right? You cannot have 21 left-handed and 21 right-handed spirals. They have to be different numbers. Whether like, you know, they're not always the same. For example, like you can always also find sunflowers which have like 34 and 55 spirals, right? So they're not always the same. You always have mutants. And also like maybe another question, so like, like while often you get Fibonacci numbers, definitely there are cases where you don't get the exact Fibonacci number. So this is like a few percent, in a few percent of the cases, they will not be exactly Fibonacci numbers, but you're gonna get like a different number, right? So definitely the process is not perfect, and you can get mistakes, right? So, and this is quite common in biology, like, it's quite robust, but it's never perfect, which is also useful because, sometimes, like you may need to adapt to the new conditions. Okay, so maybe let's move on with the presentation to also talk about the lung formation in the last 10 minutes or so. Okay, so now we're going to talk about the branching of lungs, which was done together with Celeste Nelson. Right? So here we studied the development of lungs in mouse embryos, right? And so these are snapshots of the lungs in mouse embryos. So this was 11 days old embryo, 12 days, 13, 14, 15. You see that you start with a very simple wishbone structure, but over time you start developing new branches via either bifurcations, where the existing branch splits into two daughter branches, also called the main branching, where an existing branch forms like a side branches, and so on. And this formation of new branches is very reproducible in both space and time. So if you look at the 12 days old embryo, it's always going to look like this. And what I mean, and then like, so the sequence in which these branches occur is very reproducible from one embryo to another. And the relative position of these new branch relative to the whole branch will be very similar from one embryo to another, but maybe the overall size of the lungs may be different, right? And in order to study this, what they do in Celeste Nelson, this was done by Katie Goodwin, like a grad student. So they can cut out the lungs and they can keep them alive, and they will grow, they will continue to grow and branch for like another few days before they completely die, right? As long as you keep them in the right nutrient environment, right? And in this study that I'm referencing here, we wanted to understand the process of how initially, like when there was no branch here, like you start forming like a new side branch, right? Okay, so first we need to know what are the important players, right? So what's actually growing and branching in lungs is this like a single layer of epithelium cells, which is growing and branching. Inside the epithelium is like a luminar fluid. And then the outside of the epithelium, this outside region, so this is like an epithelium. Outside region is called like a mesenchyme. And it's this mesenchyme that provides like all of these like chemical signals to epithelium where it should grow and branch. And then what's going to happen, and then it's also going to affect the epithelium through mechanics, because part of the surrounding mesenchyme cells, they will differentiate and they will form these like red smooth muscles, which you see here. So they form, these stripes are called smooth muscles, and these smooth muscles are stiffer than the other two tissues. And furthermore, these smooth muscles, they can, passively affect these by just not growing slower than epithelium. But in fact, smooth muscles, they can also actively constrict and squeeze these things, right? So both effects are possible, right? So a few years ago, like in Celeste Nelson group, they studied a bifurcation of this branch. And what they noticed is that under normal conditions, when you have bifurcations, you start developing smooth muscles at the tip of the location of the bifurcations. These smooth muscles will completely wrap around the branch and split this branch into two daughter branches, right? So under normal conditions, you get like one new branch. But interestingly, what I saw that if they put drugs, like if they use drugs for these lungs to try to prevent the differentiation of these smooth muscles, what they see is that they get these little buckles, and these are actually, I will try to show you later, these are wrinkles, right? Because what's happening is you have like a layer of epithelium that's growing with respect to the surrounding mesenchyme, and because epithelium is growing faster than the mesenchyme, then the film will affect, the epithelium will be compressed and it's going to buckle. And I'll show this later, right? So in the study that I was involved in, like I said, we wanted to look at the formation of the new side branch, right? So again, what they did is like when the embryo was 11 and a half days old, they extracted the lungs from the embryo, And what they saw that initially there were already some smooth muscles that were like located above and to the left of the location of the new branch. And then over time, what they saw that new smooth muscles formed in order to completely wrap the new branch, right? And under normal conditions, you will get like one daughter branch here. But again, if they use some drugs to try to prevent further formation of smooth muscles, so if you just stay with these pattern of smooth muscles, then you start seeing like additional branches at like down the stream and also on the other side. And I'll try to show you that we think that this is coming from the buckling instability, the wrinkling instability that we already talked about. We can also, so definitely now you see in both of these cases that formation of smooth muscles is important from proper development of new branches. And again, where these smooth muscles appear is related to the chemical signals they're coming from the mesenchyme that I mentioned these like earlier in the presentation, right? So we can also look at where the cells are dividing. So both the black and white and the colored images are the same, and they're just kind of trying to highlight in red where is the epithelium, and then where is the mesenchyme inside. And everywhere you see like a white spot, this white spot indicates a cell that was dividing at a particular instant of time. So this is the location of the new branch, and obviously you see like a lot of white spots here. So this branch is growing, right? But there's also lots of white spots above and below, and also in the mesenchyme, right? So it seems that the whole region is growing, and it's not that just the branch is growing more than the other regions around. So what you see is that this side of the lungs has more white spots than the other one, right? So this part of the lungs is growing more than the other, but the growth is quite uniform in this region, right? So we wanted to, and we can also quantify these growth profiles. So I'm going to skip this in the interest of time. So we wanted to explore in simulations what is the relative importance of the formation of smooth muscles versus these growth asymmetry that this side grows more than the other, but there's no localized growth, so that such that like we have like a uniform growth along the whole region. So I'm going to show you some simulation. We're going to start with this state, and then in one case we're just going to prescribe this non-uniform growth, and in the other case we're also going to develop these new smooth muscles, right? So so basically what will in this simulation what we do. So here the color shows the stiffness map. So the red are the stiffest smooth muscles. So these are the initial location of smooth muscles. The gray is the mesenchyme, and the pink is the epithelium. And here, for simplicity, we only assume that the epithelium is growing, and the mesenchyme is not growing. This is approximation because both are growing, but what really carries is just the relative growth of epithelium with respect to the mesenchyme. And just like what we observed in experiments, I'm going to prescribe that the growth is uniform along the z-axis, but the growth is different on the right side than on the left, right? So it's going to have some theta dependence as we go around these cylindrical coordinates, right? And then I'm just going to prescribe additional growth, and then I'm going to minimize the energy because the growth is very slow. So the system is always in quasi-mechanical equilibrium. There's no inertia, right? So now what you see when we start the simulation, you see that the tube starts to bend, which is actually also what we see in experiments. And this is because this side is growing more than the other. That's the same as I was mentioning at the beginning. But what you just noticed that at the end, this epithelium layer buckled, and now you start seeing these wrinkles. And this is because epithelium is growing faster than the mesenchyme. So epithelium is effectively being compressed, and this compression is getting released by these wrinkles. And we think that that's why in experiments where they use drugs to prevent new formation of smooth muscles, you start seeing these additional branches where it's coming from the wrinkling instability. And then now, if you try to actually mimic what's happening in experiments, that over time you start forming these new smooth muscles. So if I now do the same simulation, but now over time, you're going to start seeing more and more red here, which means that you start forming new smooth muscles, which is making like a stiff layer of muscles around the growing epithelium. What you see that in this simulation, now these stiff smooth muscles will completely suppress the wrinkling instability here. And the only way where the new branch can escape is through the narrow gap, which happens because in the narrow region, we did not form the new smooth muscles. And the reason why the new smooth muscles were not formed here is again related to the chemical signals that are coming from the substrate, from the mesenchyme, And furthermore, then there could be like a positive feedback, because once this branch is protruding, it can, this leads to high distortion of the surrounding tissue. And if the tissue can sense that it's stretched, then it can suppress then it can further suppress releases chemical signal that will further suppress the formation of smooth muscles, right? So with this, I'm coming to an end. And if you are interested in this topic, then I would like to invite you to check out the course that I developed. So the course is titled Lessons from Biology for Engineering Tiny Devices, where I talked about many of the things that I presented today, and also like many other topics. So they're available on my website, so you can find the slides. And you're also welcome to contact me via email if you have any questions about the presentation or about anything else. So now let me take a few more minutes to ask some other questions. Okay, so there's some more questions about the Fibonacci spirals. So maybe let's skip this. Do we have an explanation of cell sorting in embryonic development? Right, so the cell sorting during the embryonic development, the main idea is the same as I was showing you for Hydra, right? So it really depends on how strongly these cells stick to each other, right? So first of all, like they have, like they have to bind moderately to each other so that they can move around, so they can like it be like more like a fluid, like viscoelastic fluid rather than a solid. And in development, what's happening is like there's like a lot of shape transformations, and like a lot of times tissues are a bit fluid so that they can like deform and move to other places. And then it's really just what matters is like the stickiness of how strongly cells stick to other cells of the same type, or how strongly they stick to cells of the other types, to see whether they're going to get separated from the other groups or they're going to stay mixed with other groups. Does the branching of lung always form the same pattern, or is it unique to each individual? Is it linked in any way to chaos theory? Okay, so it's very robust, it's very reproducible, and this is what I tried to mention for mouse, is basically at least on a high level, like on the primary branches, the structure is very similar. Once you go to very tiny alveoli, there could be some differences from one person to another, but the global structure will always have like the same tree topology, right? And it's very reproducible from organ, and it's also quite similar. For example, like what we're seeing here in mouse is also like very similar to what you would see in human lungs, like the primary structure. But from one from one mice to another, or from like one human to another, like there are like lots of similarities, but definitely there are differences between the different species, right? Like I mentioned, that like the lungs in birds are quite different than the lungs in mammals. And so definitely there is no chaos theory there. How is the interaction of the microscopic level facilitated to give rise to Turing patterns, right? So I'm not really sure what is meant by interaction at the macroscopic level, but definitely like the Turing patterns can happen both on the microscopic scale, on the scale of the cell, or also on the scale of the whole organism, as we saw in like these stripes and spots of zebra. And then like basically what is important is that these chemicals are diffusing and reacting on the in this system. And this can happen like also like on the macroscopic scale. It does not have to be only on the microscopic scale. Let's see. Things are moving around. Let's see what. Floral. Why does it form this sort of floral structure with more time? Why would it remain largely circular? Does this structure have any correlation with the shape formation of flowers from buds? Okay, so I think the question is about the biofilms, right? So why do they form this flower-like structure over time, right? So the reason why they form flower-like structure is that once they delaminate, you see that this region slows down, right? Whereas the other region expands faster. So this then looks like a petals of the flower, right? And this is just coming from just the physics of how these patterns grow. One thing is that once they delaminate, maybe they lose the access to nutrients. And there could also be other regions because now maybe they're growing vertically rather than in plane, right? So there definitely could be, there are physical arguments for why they look like flowers, but the mechanisms for flower-like biofilms are quite different than the real flowers, right? So maybe since we're already like 5 minutes past the hour, maybe let's stop here. And let me thank you for your attention. And then I will be happy to address further questions later via email. So I hope you have you enjoyed the rest of the summer and the rest of the lectures of this biophysics summer school. Thank you and goodbye.